All right. Okay, so we left off here last time, and this was the diseases related to the deficiency of the carnitine, transferase, or translocase. The inability to synthesize the carnitine will contribute to the development of autism in some people. The carnitine deficiency symptoms range from mild muscle cramping to severe weakness and even death. Predominantly affects the muscle, kidney, and heart. Carnitine acetyltransferase deficiency symptoms include the muscle weakness during prolonged exercise because the muscle relies on fatty acids as a long-term energy source. These diseases, again, show impaired flow of the metabolite between the cellular compartments. This can lead to pathologies. Interesting little side factoid, many of the mental illness diseases and autism and many of the ones that are brain related, like decrease of function over time due to aging, apparently have a link to pH and similar genetic links. So they may be much more tightly related. Study just released within the last couple weeks, by the way. Kind of an interesting little side note. So the beta oxidation pathway is going to consist of 4 steps that are going to be repeated. We have step 1, which will be the oxidation by FAD. Step 2 is the hydration. Step 3 is the oxidation by the NAD+. And step 4 is the thiolysis by the coenzyme A. Each repetition is going to shorten your fatty acid chain by 2 carbons. This is called the beta oxidation pathway because the oxidation will occur at the beta atom, which, remember the alpha beta terminology from our organic chemistry? That terminology is showing up again. By the way, fatty acid naming in general uses that terminology heavily because it was developed when that terminology was popular. Okay? So you're going to see a lot of like omega fatty acid, alpha, beta, delta, that sort of thing. And again, those mean the distance from functional group, right? This right here again is that reaction sequence. Can you guys even read that? Not really? Okay. Okay. Let's try to. Better? Okay. Not a lot more I can do about that unless I cut this in pieces. So we'll leave that for now. Oh, actually, I know what I can do. I can't cut in pieces, but I can do it very quick. Okay. Yep, okay, I did do it right. Okay, can you see it now? Fantastic. Okay, this is overlapping here. Okay, normally I do a little bit better job of that, but I didn't realize that would be that hard to see. Probably partially due to the lights, I guess. Okay, so this is the acetyl-CoA right here, and again, this would be the CoA. This right here would be the what's normally considered acyl portion or acetyl portion, right? Specifically the carbonyl, which is your functional group. This then is the alpha. This is then the beta. Everybody okay with that so far? Fantastic. And so we're going to have the FAD. This is going to be your electron transporter, right? So we're going to undergo oxidation with that FAD transporting electrons away. This is then going to give us the trans delta 2 enol, CoA. Now notice again, this is right here a trans double bond due to that oxidation. Everybody okay with that so far? Okay. The delta 2 actually refers to this linkage right here. That's where that's coming from. And then enol, double bond, ene, next to the carbonyl, right? Enol. Okay. Specifically, that's a conjugated bond. Remember that? Fantastic. Okay, so what's going to happen next is we're going to have hydration across that double bond. Remember that double bonds are actually quite susceptible to hydration, specifically a double bond next to a carbonyl like this, which is going to pull electron density away, right? And so we're going to get this water attacking across. The OH is going to go on this carbon because of that resonance right here, okay? And that's then going to put the OH there. One of the H's goes onto this one. This is an L group, by the way, because remember that it's going to have 4 different spots on here, and so that's going to give it chirality. Okay. This is the third, 1, 2, 3, right? And that's going to be hydroxy acyl. This is the acyl. Okay. And that again is attached to CoA. Now we're going to get another oxidation. Remember, this process is going to be oxidizing away at these fatty acids, okay? Just like you would oxidize away at, say, a sugar or anything else to get the energy out. And so this is going to have NAD plus be the electron carrier this time. Notice the difference here. We have FAD here, NAD plus, okay? This will greatly help you in remembering this process of 4 steps, okay? It's even alphabetical. F before N, right? Okay. I know, the world helped us. Okay. So this is going to be the L-hydroxyacyl-CoA again. We have the oxidation happening. Again, this is just the same step here. Okay. And then this is going to give us our 3-keto-acyl-CoA. Notice what we've now made over here. We made a new carbonyl, right? Okay. Anybody know why that would be important? What's over here? Carbonyl. Yeah, so we're going to end up splitting this off and attaching a new CoA to it, right? And so this is going to be highly reactive, by the way, because you have 2 carbonyls right next to each other. What's going to happen is we're going to have a thiolysis. Lysis, right? We're going to split this fatty acid by shortening it by 2 carbons by attacking with a coenzyme A, CoA, which has a thiol group on it. And so this thiol is going to attack at this carbonyl here. Okay? And this is going to cause these to split off, giving us the acetyl-CoA right here. Notice the difference in the terminology. Acyl, more than 2 carbons, right? Acetyl, 2. Okay? And the acetyl-CoA you may recognize because that's going to go in the citric acid cycle, right? Fantastic. Okay, very good. So again, these are the principal reactions in your fatty acid oxidation. And again, we have the activation here, transport, first oxidation, hydration, second oxidation, cleavage. These last 4 being the 4 that are right here that we just went through. These being those ones we had led up to this, right? So the oxidation of your beta carbon is going to catalyze by the acyl-CoA dehydrogenase. This is going to generate the trans delta 2 enol CoA and the FADH2. Note, by the way, the FAD and FADH2 are going to be enzyme bound. They are a prosthetic group on this. The FAD is going to again be your electron acceptor because the delta G for this reaction is actually insufficient to drive the NAD+. This is actually why you can't use NAD+ here. Okay? Just like that previous case we looked at. We just don't have enough energy to drive an NAD+. So the electron's going to travel from the FADH2 to another flavoprotein. This can be called the electron transferring flavoprotein, or ETF. That ETF is going to donate electrons to ETF ubiquinone reductase, which is going to be an iron sulfur protein. This is going to reduce the ubiquinone to ubiquinol. Ubiquinol is going to deliver the electrons to the respiratory chain. And you may notice that essentially what we've done is we just joined it into the Q pool, right? Yeah. Everybody see that okay? That'd be normally your Q pool, right? We're just side routing it, okay? And then that just joins your normal respiratory chain with the rest of the Q pool, okay? And ubiquinol was the reduced form of ubiquinone, which is the Q, right? So QH2. And this is this right here. Again, that's that oxidation. This is the FAD right here. The FADH2 is going to then transfer to the ETF FAD. That's going to be the next carrier. This is going to transfer off onto our iron sulfur complexes, which you may remember from the previous respiratory chain, right? And then that's going to transfer off onto our ubiquinone, Q pool. The hydration of your enol CoA, so the hydration of that trans delta 2 enol CoA by the enol CoA hydratase is going to yield our L3 hydroxyacyl CoA. This is that overall reaction. We had actually seen that in the pathway before. That's the second step. The hydration is going to be stereospecific. This is why you only get the L isomer of the 3-hydroxyacyl CoA, and that's why that's formed. Remember that basically because of amino acids being almost all having chirality, enzymes basically all have chirality, and so the reactions that they function on essentially all have chirality, okay? It transfers down and through, right? So the oxidation of our L3-hydroxyacyl CoA by the L3-hydroxyacyl CoA dehydrogenase, I know the naming is just wonderful, but incredibly repetitive, which is beautiful. This generates the 3-ketoacyl CoA and NADH. And so we get rid of our chirality, we get rid of our isomerization. We're going to have enough potential energy to go ahead and push NAD plus to the reduced form, and we'll generate the 3-ketoacyl CoA. That's the one with the ketone on the next carbon. The enzyme is going to be specific for the L isomer of the 3-hydroxyacyl CoA. So if you ended up with R isomer in there, it's not going to function. The thiolytic cleavage of the 3-ketoacyl CoA by the beta-ketothiolase yields the acetyl-CoA and the acyl-CoA, which have 2 carbons shorter. Note, by the way, that the naming of this is incredibly nice because it's beta-ketothiolase. See what that tells you? Which carbon are we acting on? It's 2 carbons down from the previous carbonyl, right? Which is beta. It's a keto group. A thiol is going to attack it, and ACE for enzyme, right? So it actually tells us everything about the reaction again. Again, we've lost 2 carbons. We end up with the acetyl-CoA and the acyl-CoA. And that acyl-CoA can then feed back in, and we can do this all again, right? Is this a cycle, by the way? No, it isn't. And the reason it isn't is because we don't actually get the same group coming back in, do we? We get 2 carbons less. I know, right? So it's not technically a cycle, okay? But yeah, it has the same reaction happening to that same thing minus 2 carbons every time, right? And so it's very much analogous to our glycogen, right? Where we're chopping away. Our shortened acyl-CoA, this undergoes another cycle of the oxidation, starting with the acyl-CoA dehydrogenase reaction. So the long-chain acyl-CoA dehydrogenase is going to oxidize fatty acid chains containing 12 to 18 carbon atoms. the medium chain acetyl-CoA dehydrogenase, again, oxidase fatty acid chains that contain from 4 to 14 carbons. The short chain acetyl-CoA dehydrogenase oxidizes only 4 and 6 carbon fatty acid chains. And so these are the definitions. If you see long chain, short chain, medium chain, these carry over the rest of the way. Okay? So this is typically the definitions for that. The enyl-CoA hydratase, hydroxyacyl dehydrogenase, and the beta-ketothiolase act on the fatty acid molecules of any length. And so these part of that process, they don't matter how long it is. It has to be more than 2 carbons, because if it's 2 carbons, what's going to happen? If it's 2 carbons, what would it be? Yeah, it'd be acetyl-CoA, right? You don't need to do this. It can't actually be 3 carbons either, and we'll talk a bit about that when we get odds versus evens. Because if it's 3, you would actually not be able to do this reaction right here, right? See that? This would be an end one. This wouldn't be a secondary. Okay? Okay, so each round of your fatty acid degradation is going to remove 2 carbons. And so right here we have it nicely color-coded, so you can see that better. Can everybody see the color coding? Okay? Or at least tell the difference? Okay. And so we get these red here. These are the first 2 carbons. Again, we get acetyl-CoA Z off each time. And there's the next one. And there's the next one. And we keep going. Okay? So the reaction for 1 round of beta oxidation would be this overall reaction here. The complete reaction of, for example, palmitoyl-CoA, this is a C16 fatty acid, would be this right here. And so we have this process happening how many times for a C16 reaction? I made it go away. So how many times would the beta oxidation occur for a C16 fatty acid? Exactly, yeah. You divide it by 2, right? And then minus 1 because the last one doesn't need to do it. It's perfect. Got it. Like that, right? And so we do it 7 times, but we actually get 8 acetyl-CoAs out, don't we? Because technically we started with 1 of them, didn't we? The process of processing of your NADH, FADH2, and the acetyl-CoAs in the cellular respiration would generate 108 molecules of ATP. This would be an approximate number. But 2 ATP molecules are actually consumed in the palmitate activation, that first reaction that we started this with. And so we would get a net yield of something along the lines of 106 molecules. Remember, though, that individual organisms will have slightly more and less efficient pathways. Okay? So this is a good approximation number. Right? Now, the beta oxidation alone cannot degrade unsaturated fatty acids. When monounsaturated fatty acids are degraded by the beta oxidation, your cis delta 3 enyl CoA is formed. This is not a substrate for the acetyl-CoA dehydrogenase. So the cis delta 3 enyl CoA isomerase would convert the double bond into a trans delta 2 double bond. This would then yield a trans delta 2 enyl CoA. This is your normal substrate for your beta oxidation, and so you can continue chopping away. Okay? The unsaturated fatty acids with odd numbers of double bonds require only the isomerase. And so this is what we're talking about right here. So this chain right here, see how it's C7 here? We'd be cutting. We'd encounter this eventually, right? So these are just normal beta oxidation reactions up here. Eventually, we're going to get the cis delta 3 enyl CoA. Now, can you make a trans bond here? Or even a carbonyl for that matter? No, we're stuck, aren't we? Yeah. So if we were just trying to do the enzymes in the regular beta oxidation, we're shit out of luck. We're stuck, aren't we? Yeah. We just don't have the ability to do that anymore. And so what's going to happen is we have the enzyme that's the cis-delta-3-enyl-CoA isomerase. This is going to help do a rearrangement. We'll rearrange this cis double bond into a trans double bond right next to it. Notice the delta numbers, delta 3, delta 2. Everybody see it okay? Okay. And that's literally your offsets. So 1, 2, 3 starts there. See that? This one being 1, 2 starts there. Okay. Does anybody have any questions about that so far? Okay. Fantastic. Okay, so when the polyunsaturated fatty acids are degraded by a beta oxidation, your cis-delta-3-enyl-CoA isomerase is also going to be required to convert the cis-delta-3 double bond into the trans-delta-2 double bond. The acetyl-CoA is going to produced is going to contain a cis-delta-4 double bond, and its dehydrogenation is going to yield a 2,4-dienyl intermediate. The 2,4-dienyl-CoA reductase is going to use NADPH to reduce the 2,4-dienyl intermediate to the trans-delta-3-enyl-CoA. The cis-delta-3-enyl-CoA isomerase converts the trans-delta-3-enyl-CoA to the trans-delta-2-enyl-CoA, which is the normal substrate for your beta oxidation. So we're going to be adding some extra steps here in just a moment. Your unsaturated fatty acid with even numbers of double bonds is going to require both the isomerase and the reductase. You'll need both. And so this is that right there. See how it's polyunsaturated, 2 unsaturations here? And they're near each other? Okay. And that's linyl-CoA, by the way, which is linoleic acid, in case you're wondering. That's the cis-delta-3-enyl right here. CoA isomerase is going to act on this. And what needs to happen is we need to rearrange these. Do you remember your conjugated double bonds here? And what sort of activity those things have? Anybody remember that from Organic 1? No? Okay, so these are conjugated, right? Because they're the pi bonds up right next to each other, and so they can exchange electrons in them. Okay? If we were to try to react one of these, that could transfer electrons all the way through that system, right? And so we could get side reactions where it would not make the acetyl-CoAs we're trying to make, right? And so what's going to happen is this is going to break that conjugation system and rearrange it in such a way that we can do our attacks normally, okay? Okay, i.e. that we're going to end up making one of the normal products, right? And so right here, this is that cis-delta-3-enyl-CoA isomerase. It's going to rearrange this cis bond into that trans bond just like we saw before. That's that same one we saw the pathway for before. And then what's going to happen then is we'll again go to the next step, and this is going to end up being a splitting this off right here. See how this is going to get taken off? Okay. And then we're going to be left with this right here. Notice we're right next to this double, this right here. See that? We still have that carbon left from the blue. Okay. And that's now our carbonyl. That's the normal beta oxidation pathway. And so now we're one less for our two, we have lost 2 carbons from the previous beta oxidation. What's going to happen next is we're going to have the acetyl-CoA dehydrogenase. This is going to then react, this right here, and give us the trans form of this one right here. Okay? And that's the 2,4-dienyl-CoA. Now, notice again, we have reestablished a conjugate system, haven't we? Yeah. And so we're going to have to use the 2,4-dienyl-CoA reductase. what this is actually going to do is it's going to take that conjugated 2 double bonds, and we're going to reduce that. Okay? Note, by the way, it's kind of an interesting opposition here, because this overall pathway is oxidations, right? And we had to reduce it so we could do the oxidations. Okay? But anyways, the 2,4-dienyl-CoA, again, that's the 2,4-dienyl-CoA reductase for the enzyme. Again, we're going to use the NADPH. This is going to be our electron carrier to do this reduction with. Okay? And what we'll end up left with is the trans double bond in the correct position to do the next beta oxidation reactions, okay? Which is this form right here. Does anybody have any questions about this? No? Don't forget the last rearrangement right here. This can go back and forth, by the way. It's an equilibrium step. And that actually gives you the trans delta 3-enyl-CoA in the proper positioning. Okay. So your beta oxidation of your fatty acids with odd numbers of carbons is going to generate propenyl-CoA in the final round of your degradation. The propenyl-CoA, that is not acetyl-CoA, is it? Propenyl means it has how many carbons? Pro is 3, right? Yeah. And so, can you do 2 beta oxidations on that? No. And you don't have acetyl-CoA, do you? No. Oh, shit. We're out of our damn pathways again, aren't we? So we need to have something to fix this. So you're going to have an activated 3-carbon unit in the propenyl-CoA. This will be converted to the succinyl-CoA. Succinyl-CoA can then enter the citric acid cycle. Yeah, that's the same succinyl-CoA. You remember the succinate, etc. Okay. This can be converted into the oxaloacetate to provide carbon for your glucose biosynthesis as well. And so this actually pulls double duty. Your propenyl-CoA carboxylase, this can be a biotin enzyme that carboxylates your propenyl-CoA. This will yield your D-methyl-malonyl-CoA. This step actually does require ATP. The mechanism is similar to the homologous enzyme pyruvate D carboxylase. Your D-methyl-malonyl-CoA is racemized to the L-methyl-malonyl-CoA. Your methylmalonyl-CoA mutase is going to catalyze the isomerization of the L-methyl-malonyl-CoA. This will give you then the succinyl-CoA. This will contain a derivative of the vitamin B12, cobalamin, and its coenzyme. All right. So again, this is that overall reaction pathway. Here we have the propenyl-CoA. You can see the 3 carbons here. And so notice this is extremely analogous to what we've seen before, where we add a carbon on there to fix this, so we get more carbons, right? And so what we're going to have here is carbamate, which is our typical sort of source of carbon, right? Remember, that's the acidic form of CO2. This is going to be plus an ATP. We'll use that ATP energy to do this process. And so that's going to go to the ADP and an orthophosphate. That will give us our D-methyl-malonyl-CoA. Again, that's because of this group right here. See that carbon right there? Okay, and again, that's the methyl-malonyl. Methyl right there, malonyl. This is then going to have a series of equilibriums, which will allow for the rearrangements of this. This is because these are fairly similar energies for these different steps. So here we're going to have the conversion to the L-methyl-malonyl-CoA. Again, we've just swapped these 2 groups, right? See that? And so we flipped the stereochemistry here. And then this is going to be able to go, the L-methyl-malonyl-CoA will be able to go to the succinyl-CoA. And so this is, again, that succinyl-CoA that you see in the citric acid cycle, etc. Okay? one of those ubiquitous molecules, right? That we can go into quite a few different pathways from. Cobalamine enzymes are going to catalyze the three types of reactions. We have the intramolecular rearrangements, the methylations, the reductions of ribonucleotides to the deoxyribonucleotides. The coenzyme B12 is going to be required for only 2 mammalian reactions. We have the conversion of L-methylmalonyl-CoA into succinyl-CoA and the formation of methionine. The vitamin B12 is only made by prokaryotic organisms. Here's the core of cobalamine. It's a core and ring with cobalt atoms. Here's the core and ring right here, by the way. And then here's the cobalt atom right there. Okay? Okay. And that's that. Anyways, anybody have any questions on that? Okay. By the way, do you need cobalt in your diet? Yes. Did you realize you needed cobalt in your diet? No, that probably never occurred to you, did it? Yeah. It's surprising some of the trace minerals you need in your diet, right? Technically, you can get this from eating other eukaryotic organisms. But there are certain dietary restrictions that are happening in the world that do not naturally get these. Okay? Or get these at very low levels. Right? And so actually have to be quite careful. Yeah. So be careful if you do any weird diets that you don't have like moderation. Okay? Keto, but veganism is another one that has a lot of problems. Funny enough, keto usually gets screwed up less often than veganism. But both are very much diets that people get on and have no idea what the fuck they're doing and then screw that up horribly, right? And cause them serious problems. So the rearrangements catalyzed by coenzyme B12, these are going to be exchanges of two groups that are attached to adjacent carbon atoms of the substrate. Coenzyme B12 will serve as a source of free radicals for the abstraction of hydrogen atoms. And so right here we actually can see an exchange of a hydrogen atom and the substituent next to it. Could be a methyl, it could be whatever. Okay? This is the homolytic cleavage actually of the carbon-cobalt bond of a 5' deoxyadenosine cobalamine. It's going to generate the CO2 plus form of the coenzyme and the 5' deoxyadenylyl radical, which is this right here. It's that CH2 radical. But remember, that will still be attached to the deoxyadenylyl. Okay? And so this is actually this right here. Again, we attach the CH2 to this cobalt. It's in the 3+ form because it's going to then have a homolytic cleavage, meaning the bond breaks evenly across. We'll leave one electron on the carbon, one electron on the cobalt, which will bring it to 2+. Okay? And that will generate a free radical carbon. I know, you guys probably thought free radical carbons were never going to show up in my biological stuff, right? Well, guess what? Yeah, it's funny how many of these weird reactions that you were probably like, I don't need that for biology. Yep, free radicals. The radical abstracts a hydrogen atom from your substrate to form the 5' deoxyadenosine and a substrate radical. The substrate radical spontaneously rearranges, producing a different radical. Remember, these radicals are highly, highly reactive, okay? These things are going to have rearrangements all the time. Remember, in organic chemistry, we had those damn methyl shifts and all sorts of activities going on, right? Rearrangements where the radical was going on oxygen, etc. Okay? So the radical is going to abstract the hydrogen atom from the methyl group of the 5' deoxyadenosine. This will yield the succinyl-CoA and regenerate the 5' deoxyadenylyl radical. So this is what that reaction looks like. Here we have the L-methanylmalonyl CoA, and here we have that 5 deoxyadenylyl radical right here. And so what you're going to see here is it's going to react to that methyl group right here, which is fairly exposed. This will regenerate your 5-deoxyadenosine and give you a free radical on this, which was your methylmalonyl, right? Once that has that free radical right there, well, this can very easily rearrange by pulling electron density, like so. And so what we're seeing here is that rearrangement. Can everybody see that okay? Okay. So we swapped bonds. See, that was here. Now it's here. And that only happens because you have a damn open p orbital with free radical in it next to it, right? So those pull bonds very easily. Notice that's very similar to the methyl shifts we showed you back in Organic Chemistry 1, right? Yeah. All right, so your succinyl-CoA is right here, and now we've generated that. Note that we are going to take the extract the hydrogen from that 5' deoxyadenosine, regenerating the radical form. And then now that goes. By the way, like many free radical reactions, what sort of reaction is this? It's a chain reaction, isn't it? Yeah. We actually regenerated this, right? And so this is going to be a propagation step. Isn't that cool? Man, that was like the original reaction I showed you, right? In organic chemistry, and we're all the way back into it. We just wrap around every time, don't we? Okay. So the active sites of the methylmalonyl CoA mutase is going to reveal how the radical formation is actually accomplished. And so right here we have the methylmalonyl CoA right here. Again, that's the sulfide thiol linkage right here. This right here is the 5-deoxyadenosine, and then here we have the cobalamine. Now note again, we want this to be able to react right away, because if this had to diffuse first and then react, what would happen? Is a radical going to nicely diffuse in water with a bunch of reactants around it? No, we would get all sorts of side reactions, right? And most of those would probably be termination reactions, like you saw in your chemistry and organic chemistry, okay? But you'd generate a hell of a lot of free radical products, wouldn't you? Many of them probably would be poisonous. So it's going to have it right next to each other. And so right here is actually the bond that's going to get cleaved. And you can see it's just going to come in here. It's going to generate that free radical. And then the thing that's right next to it to react, the hydrogen is going to abstract, right? And so they're right next to each other the whole time. Okay. Anybody have any questions? This is going to be part of that stabilization and helping make this reactive right here, by the way. Is remember that we're having this so it can stabilize that charge on that cobalt, right? Okay. And that's part of what that crown is going to do. Anyways, we could go into that all day, but we'll keep going. Some of the enzymes activities are actually just fascinating. Okay, so we have peroxisomes. These are going to be a small membrane-bound organelles present in most eukaryotic cells. The oxidation of long-chain and branched fatty acids is going to take place in the peroxisomes. The function to shorten very long chains to make them better substrates for the beta oxidation in your mitochondria. The oxidation will halt with the formation of octanyl-CoA. So you may have been wondering, some of these chains seems like they'd be really long and make life really difficult to move into that mitochondria, right? What the hell do we do? Does it make sense to move around like a 20 trailer long with your car? No. So we split those up, we move them in, we do the reactions, right? Okay. And so the flavoprotein acetyl-CoA dehydrogenase is going to transfer electrons to the FADH2, then to the O2. This will yield then H2O2. What's H2O2 again? Hydrogen peroxide. You got it, yeah. So we make this fairly reactive intermediate, don't we? The peroxisomes, and this is where it gets its name, by the way, peroxisomes, peroxide, is going to contain high concentrations of your catalase. This will help to degrade the H2O2 into your water and oxygen. Subsequent steps are going to be identical to the beta oxidations, but are carried out by different enzyme isos forms. So we have different variants of those enzymes. And so right here, this is the initiation of your peroxisome fatty acid degradation. Again, it's going to require that flavoprotein. And so here we have this very long chain. And so our acetyl-CoA dehydrogenase in the oxidized form with FAD right here, again, it's the oxidized form, FAD, is going to then take that, it's going to oxidize this bond. This is going to give us the reduced form, and this would give us further oxidation down here, right? Now, notice this will generate the hydrogen peroxide here when we go from the acetyl-CoA dehydrogenase, reduced form, to the oxidized form. This is going to reduce your oxygen to the reduced form, which is the hydrogen peroxide, partially reduced form, I should say. We're going to use the catalase to further reduce this, and then get this oxygen back in the O2 form here. Now notice that overall we are doing those reductions. We're gathering them on one oxygen, and the other oxygen is going to the more oxidized form. Everybody see that? Isn't that neat? So do you know what we're doing here? Which is the reducing species in this reaction? Oxygen. Which is the oxidizing? Oxygen. we're oxidizing one and we're reducing the other of the oxygens. Isn't that funny? Yeah. So at this point, they're both plus ones, right? They would have been 1, 1. This one, they would be 2 reductions for this. This one has lost one of its reductions to go to an oxidation, right? Ooh, ah. Oh, you guys just don't appreciate that, do you? That's a little bit sad. Okay. So again, this would just be further oxidation like your normal beta oxidation pathway. The vegetable oils are going to be rich in polyunsaturated fatty acids. These are going to be unstable. They're going to be readily oxidized. Your polyunsaturated fatty acids, these may be hydrogenated, converting them to saturated and monounsaturated fats. This causes undesirable isomerization of some cis double bonds, yielding trans fats. The saturated fatty acid and trans fat consumption promotes obesity, type 2 diabetes, and atherosclerosis. The evidence suggests that they slow the beta oxidation, directly promote an inflammatory response, and mute the action of insulin and other hormones. And this is really funny, actually. So if you ever want to see something really like stupid for health decisions, but kind of ironic, look at the history of McDonald's fries. Okay? It has this entire history where they actually started with something healthy-ish for French fries, and then they've repeatedly had these boycotts and bans and health-concerned people giving them problems, and it has actually made the fries worse for you, and worse for you, and worse for you, and worse for you, repeatedly over 40 years. And so now they're almost the worst thing that you can eat, right? And it's all because of health-conscious activists, which is just mind-blowing. But it's because the food pyramid and the money that's been going into health activities for the last 40 years, about 30 of those years, were directly anti-science. Yeah, and the people that actually reported real results were blacklisted in the field. And so the government heavily pressed its thumb down on the science. Yeah. That's the food pyramid, by the way, in case you're wondering. Which, did you guys know that's coming back? Yeah, the food pyramid came back. Look at Biden's health guidance website. Yeah, I probably did. Yeah, it actually happened like a year and a half, 2 years ago. Yeah, look up Lucky Charms versus an egg. Lucky Charms versus egg.
Oh, yeah, you said that. Lucky Charms, not eggs.
Yeah. Lucky Charms is twice as good for you as eggs are.
I like Lucky Charms. I know it. I like fries. You know, such good fries and cold. I love cold. I just like Pringle cut fries. Also, very good fries.
You guys ever had the Arby's fries?
The curly ones? Yeah.
I'm not necessarily the curly fries, but they some places have something they call steak fries. They're like the curly fries, but they're straight-ish. And yeah, so they have like a really good seasoning to fry ratio.
The spicy fries, I think
They are. Yeah. The grill over there is actually a great restaurant. It's like the best restaurant in McKenzie.
Yeah, I think the city is with like the chicken one with onions and peppers in them with spicy fries.
Have you tried their new little pizzas?
No, I haven't.
Try it. They have a barbecue one. Barbecue chicken. Yeah. It's really good. They even make flatbreads too if you don't like as much crust. Yeah. Anyways, it's a great restaurant. Good prices too. I have no idea where I left off. Yeah. And now I'm a little hungry too because I haven't had anything to eat. So the acetyl-CoA, you guys could have brought some food from the grill.
I had an exam before this. I had to
pick up an exam before this class.
Shoot. Okay, so the acetyl-CoA formed in this fatty acid oxidation. This can enter directly in the citric acid cycle. If a fat and a carbohydrate degradation are balanced, the acetyl-CoA combines with the acetyl-acetate in the first step of the citric acid cycle. The oxaloacetate availability will depend on your carbohydrate availability. Now, keep in mind that because we just directly go into these acetyl-CoAs essentially directly, right? We have basically nothing else going on. This actually does create an imbalance in the cycle, okay? And so we can actually get kind of an interesting behavior, which is if fasting or diabetes, the oxaloacetate will be consumed because we'll need to make glucose for our brain red blood cells, right? And so this will go into the gluconeogenic pathway. The acetyl-CoA is then going to be diverted to form acetoacetate and your D3 hydroxybutyrate. Your ketone bodies, this is where ketogenesis, the keto diet names come from. Ketogenesis not just being the ketone diet, by the way, the keto diet. Also being things like fasting and such. But ketone bodies are going to be the acetoacetate, the D3 hydroxybutyrate, and acetone. Notice again, all those have ketone, own in their names, don't they? Yeah, and so those are ketone bodies. They're water-soluble. They're transportable forms of the acyl units. Abnormally high levels of ketone bodies are going to be present in the blood of untreated diabetics because, again, they're water-soluble, so they can go out to the blood quite easily, and you can have elevated levels then. Okay. Interestingly enough, this is also a part of the reason why, like, if you look at the urine of diabetics, they have a smell to it, right? And it has a different taste to it as well. And that actually was one of the old ways of testing for diabetes. Somebody would literally lick it. And it'll taste like a floury sweet, I guess. I have no idea. I've never tried it. Yeah. You can smell it, though, too. If I absolutely had to save somebody's life, probably. But no, that's not going to be something I just try for the hell of it. Thank you, but no. Yeah. I mean, I would try a lot of different foods. Basically, my deal is if like 4 other people are willing to try it with me, I'll definitely try it. So if I go to another culture, I always end up trying everything. As long as the other people eat with me, though. If they just tell me, oh yeah, you should eat that. No. You gotta eat with me, man. Because I've done that to people. Yeah, you should eat the raw deer heart. Go for it. Yeah, that thing's chocolate. Eat that. Yep. Acetoacetate. This is going to be formed from acetyl-CoA. This is in 3 steps. Step 1 is the thiolase catalyzes the condensation of 2 molecules of acetyl-CoA to form acetoacetyl-CoA. Step 2 is the acetoacetyl-CoA. This reacts with the acetyl-CoA and water. This is going to yield your 3-hydroxy-3-methyl-glutarol-CoA. This is typically abbreviated HMG, H-hydroxy-methyl-glutarol, and then CoA. This will also give you CoA, just pre-CoA, because you took 2 CoAs in. See that? See, free one. Step 3, this is the 3-hydroxy-3-methyl-glutarol-CoA, HMG. Is then going to be cleaved to the acetyl-CoA and acetoacetate. The sum of these reactions is actually this right here. This right here being the pathway. And so we have 2 of these acetyl-CoAs coming in. We'll lose 1 CoA, we'll get acetyl-acetyl-CoA, and then we need another acetyl-CoA. This is then going to come in, we lose the CoA, we use up a water, and this will give us our 3-hydroxy-3-methyl-glutarol-CoA right here. Okay, and then notice the wide variety of ketones we get to see. Ooh, beautiful ketones. Okay, now, the next is going to happen is we're going to release one of these acetyl-CoAs right here. This is going to give us the acetoacetate, and this is the acetoacetate right here. Notice, by the way, acetate right there. This is the aceto right there, the blue part. Next is going to happen is it can go 2 different ways. It breaks into 2 different parts here. We can have the equilibrium. This is going to take NADH. We're going to reduce this to the reduced form, which is this right here, which is the D3-hydroxybutyrate right there. And again, that's the reduced form because we reduced it across this double bond, the carbonyl. You get the alcohol there. By the way, that does give us stereochemistry, a chirality center, right? The other way would be acetone. And so this can break up and give us acetone here. And by the way, is acetone good for you? No, acetone is not good for you. And it can go into your blood quite easily, right? Okay, this is actually why people that do the ketogenic diets or fasting and don't do it correctly with the proper salts so you have the proper enzymes to take care of this, cause things like liver damage, by the way. This is your biggest reason why. Because you're mostly going to make acetone in the liver. It's going to damage the liver first. And the liver is also the one that has to work to remove that, right? And break that down. So the reduction of the acetoacetate, in the mitochondrial matrix is going to yield your D3-hydroxybutyrate. It's going to catalyze by the D3-hydroxybutyrate dehydrogenase. Your ratio of hydroxybutyrate to the acetoacetate is going to depend on your NADH, NAD plus ratio inside your mitochondria. As a beta keto acid, the acetoacetate also undergoes a slow spontaneous decarboxylation to acetone. This may be detected in the breath, and this is actually why people that are having issues sometimes have this flowery smelling breath, and you can actually detect that in them. You can actually detect this in patients in hospitals oftentimes, and nurses are trained to do that. So are most like PAs and doctors and therapists as well. Yeah. Under starvation conditions, your acetone may be captured to synthesize the glucose. So the liver is going to be your major site of production of the acetoacetate and 3-hydroxybutyrate. The acetoacetate and 3-hydroxybutyrate are going to be carried from the liver mitochondria into the blood by the transport proteins, delivered to other tissues like the heart, kidney, etc. The brain adapts to utilization of acetoacetate during starvation and diabetes. And that's why if you actually have the correct nutrients that you're taking, like you have to take a mixture of salts and water and stuff while you're fasting, you can actually be fully functional during fasting and have no problems at all. Okay? Because do you actually need to take in sugars? No. No. You can fully replace the sugars. Okay? Does it make your body carry an extra strain, and so if you don't have all the proper enzymes, you're kind of fucked? Yes. Yes. Which is why people get into a lot of problems doing this when they don't have a team of professionals surrounding them, right? So multi-millionaire, billionaire types, sure. People that want to take 18 hours a day to monitor their health levels and do this perfectly, okay. Average person that's not a YouTuber, no. No, it's a terrible idea, right? But some people can do it. So this is the pathway integration. Your liver supplies the ketone bodies to the peripheral tissues. This is your fat cell right here. You have your triacylglycerol. We're going into glycerol and fatty acids. You may recognize that. Going into the blood. This goes into the liver. Your liver is then going to take that glycerol. It can then convert that to glucose. The fatty acids are going to go into the fatty acid, acetyl-CoA, ketone bodies. This is going to be in the mitochondria. This is then going to be transported back out into the blood, both the glucose and ketone bodies. These are going to go to the parts that need it, such as heart muscle cells, renal cortex cell, brain cell, usually during starvation, but you can actually trick your body into doing starvation if you go on a ketogenic diet. Okay? Ketogenic diet is basically starvation without being starved. Okay. By the way, do you guys know what the 1890s treatment for epilepsy was? One of the most effective treatments, in fact, for epilepsy ever discovered. Starvation. Yeah. You can treat epilepsy through just putting somebody on a ketogenic diet and completely cure it. Yeah. And, well, many cases, not all. Again, that's a effect, right? Epilepsy is effect, not a cause. But you can also do the same thing for quite a few cases of things like inflammation, which can be things like autoimmune diseases and all sorts of stuff. It's crazy. But it only works in a portion of those cases because they're not all the same causes sometimes. And it doesn't completely stop whatever is happening. But you can actually treat it long term, which is pretty crazy. At least to me, that seems crazy. Ketogenic diet, boom. No more bipolar. What the hell? Anyways, ketone bodies, acetyl-CoA going into the citric acid cycle, giving you CO2 and H2O, and yay, I powered my brain. Okay. So step one, the acetoacetate is going to be activated by the transfer of CoA from succinyl-CoA. This can be catalyzed by the specific CoA transferase. Step two is the acetoacetyl-CoA. This can be cleaved by the thiolase. It's going to yield 2 molecules of acetyl-CoA. Acetyl-CoA can enter the citric acid cycle from this. The acetoacetate again, going into the acetyl-acetyl-CoA. Again, this is going to have the succinyl-CoA joining with acetyl-acetate gives you succinate, and the CoA transferase enzyme is the one that does this. The acetyl-acetyl-CoA is then going to have another CoA come in. Thiolase is going to act on it. This is going to split the acetoacetyl group into two acetyl-CoAs. And then acetyl-CoA is normally where we begin this cycle, right? And so we can go in citric acid cycle. Your 3-hydroxybutyrate, this must first be oxidized to produce the acetoacetate and NADH for use in the oxidative phosphorylation. And so again, this is the 3-hydroxybutyrate that we had seen before. We're going to be reducing the NAD plus to the NADH and oxidizing our hydroxy to carbonyl right there. Let's talk about ketogenic diets in a bit more detail. So the ketogenic diets, this is going to be a diet to promote the ketone body formation. They're used often as a therapeutic option, especially for children with drug-resistant epilepsy. It's kind of the go-to procedure now because a lot of the epilepsy drugs can have very bad side effects, especially in a developing brain, right? The diets are going to be rich in fats and low in carbohydrates with adequate amounts of protein. There is some evidence that has started accumulating, but it is very new, like 1 or 2 years for publications that are supporting this, that show that if you go 0 carbohydrates, you can actually cause the person to have a very small proportion of the people treated to actually permanently cure their condition. And you can then reintroduce carbohydrates later, which is just wild, but that is such a dangerous diet to try to do by yourself. So do not just go and do that. The recent research in mice actually suggests ketogenic diets can alter the microbiome, resulting in therapeutic effects, may extend lifespan, improve memory, maintain long-term health. This is why you see all these billionaires going on fasting routines or ketogenic diets, by the way, in case you wonder. But again, remember, when you see those billionaires doing this shit, they have a team of on-the-call doctors, okay? And they're getting, like, their metabolic pathways looked at daily or at least weekly. They're not the normal person that has to wait in line to see a doctor, right? Yeah, they don't just have one doctor on call. They have a team and a lab that will do tests, 1-day turnarounds. So the absence of insulin in patients with insulin-dependent diabetes has biochemical consequences. The liver cannot absorb glucose and cannot provide oxaloacetate to process fatty acid-derived acetyl-CoA. The adipose cells continue to release fatty acids. This results in the formation of large amounts of acidic keto bodies. Did I tell you guys they just released a publication this last week linking the pH of the brain to all these different diseases? Yeah, so supposedly the pH of the brain is a very strong predictor of Alzheimer's, autism, everything else. Yeah. So, diabetic ketosis is going to result when insulin signaling is going to be impaired. Here we have our adipose tissue. Again, no glucose coming in. Here we have our liver. Again, no glucose coming in. And so free fatty acids are going to be released. They're going to go to the liver. They're going to come back here. And your citric acid cycles can be greatly lowered, right? Because we don't have oxaloacetate feeding in. So we have to use the limited number of the ones we do have to try to do what we can, okay? And so you're going to have ketone bodies forming. These ketone bodies tend to be a bit more acidic than most of the other groups. And so your blood pH starts dropping. When your blood pH starts dropping, what's that going to do? Acidosis, which can then lead to coma and death, okay? So the acetyl-CoA cannot be converted into pyruvate or oxaloacetate in animals. The reactions that generate the acetyl-CoA from pyruvate is irreversible. No net synthesis of oxaloacetate is possible because 2 carbon atoms are going to enter the citric acid cycle and leave in the 2 carboxylate reactions. Plants and many microorganisms have the glycoxylate cycle for converting the carbon atoms of the acetyl-CoA into the oxaloacetate. And this is the real central problem of why humans don't really handle fasting well without, considerable thought process being put into it, and why we don't really handle things like a ketogenic diet well without being very careful about it. Okay? Though we can. We just need to get the correct nutrients in, right? So the fatty acid synthesis can be a complex of enzymes that will synthesize our fatty acids. Again, this is not a sugar, by the way. You don't necessarily need sugar to get the oxaloacetate. So if you're wondering how that fits in with you don't need sugar in your diet, there you go. But again, it is much more difficult to do your diet without it. The fatty acid synthesis can be a complex of enzymes that's going to synthesize fatty acids. Many tissues, such as the liver and adipose tissue, are going to be capable of synthesizing your fatty acids. Your fatty acid synthesis is going to be required under certain physiological conditions. Some of the examples of these are during embryonic development, also during lactation and mammary glands. The fatty acid degradation and synthesis steps are going to be reverse of each other in their basic chemistry. The fatty acid degradation is an oxidative process that's going to yield activated acetyl units. The fatty acid synthesis is going to be a reductive then. And this is going to begin with an activated acyl group and an activated malonyl group. And so you see they're going to be quite similar, just in reverse. Okay? Okay, so again, this is the degradation, and this is that, remember, we're starting with an activated one here. We already have the CoA on it. And this is that 4-step process that we talked about before, beta oxidation, where we split 2 carbon acetyl groups off each time. Everybody okay with that? So again, that pattern was oxidation, hydration, oxidation, cleavage, right? Okay, so if we go the reverse here, this is going to be the fatty acid synthesis. What we're going to see is that we're going to start with the activated acyl group. We're going to then take the activated malonyl group. Note that these are not direct analogs of each other. So you start with slightly different reactants here, okay? Okay, so again, you're going to do a condensation on this first step. Now, this is going to release CO2, making this an irreversible reaction under physiological conditions, okay? And this is going to then give us that condensation right here. Notice condensation versus cleavage. Putting together versus cleaving, right? They're very analogous to being opposites. Then we have a reduction where we had an oxidation. Then we have a dehydration right here where we had a hydration here. I know, this is actually quite nice, isn't it? And then we have the reduction in place of the oxidation. So other than the starting point, we actually have very nice analogous reactions, right? All basically direct overlap. It's only the starting reaction that's a little bit different because we need to drive this forward, right? Whereas this was being driven forward by the energy before. So the synthesis is going to occur in your cytoplasm, whereas degradation is going to occur in the mitochondrial matrix. The intermediates in the synthesis are going to be covalently linked to the sulfhydryl group of an acyl carrier protein, or ACP, whereas the intermediates in the degradation are actually going to be covalently attached to the sulfhydryl group of the coenzyme A. The synthesis enzymes in higher organisms are going to be joined in a single polypeptide chain, whereas the degradation enzymes are not linked covalently. The activated donor of the 2 carbon units in the synthesis is actually malonyl ACP. Remember, this is that carrier protein. Whereas the degradation releases acetyl-CoA. The reductant is going to be, the synthesis is the NADPH, whereas the oxidants are the degradation of the NAD plus and the FAD. So this is actually some major diverging points, right? The isomeric form of the hydroxyl acetyl intermediate in your synthesis is D, whereas the form in the degradation is L. The malonyl-CoA acetyl-CoA is going to be a carboxylated, yielding malonyl-CoA. This is going to be catalyzed by the acetyl-CoA carboxylase 1, which is ACC, biotin, requiring cytosolic enzyme. This is going to proceed through a carboxybiotin intermediate. It's going to require your ATP hydrolysis. The formation of the malonyl-CoA is going to occur in two steps, and these are the two steps right here. The first one is in equilibrium, which is the biotin enzyme plus ATP, and note again that's the dissolved form of CO2, if you will, or the carbonic acid. This is going to give you the CO2 linked to the biotin enzyme, ADP, and an orthophosphate, this providing the energy to do that linkage. The CO2 biotin enzyme is going to react with the acetyl-CoA. This will give you your malonyl-CoA and biotin enzyme. The intermediates in the fatty acid synthesis are going to be linked to an acyl carrier protein. The acyl carrier protein, which is again that abbreviation ACP, this is that 77 residue polypeptide with a phosphopentanthine group that's going to be attached to SER. Phosphopentanthine group is going to serve as your fatty acid attachment point within your ACP. This just serves as the fatty acid attachment point in the much smaller CoA molecules. The much larger ACP molecule can be envisioned as your sort of macro big CoA, okay? Whereas the other CoAs are your small CoA. You guys okay? Back there? We getting another leak back there? Shoot. Oh, man. I don't know what to say to you there. Yeah. Anyways, think of the ACP here as being big CoA. Okay? Macro CoA. Whereas normally in organic labs, we were doing micro. Okay. Now, in the acyl carrier protein again, this is the pantothenine group. Again, this is your coenzyme A right here. You'll notice these are quite analogous, aren't they? See that? Yeah. So again, CoA had the same sort of part right here that actually does the linkages. The only real difference here is the heads, which are CoA was small, whereas the carrier protein is a protein and quite large. These are the principal reactions, your fatty acid synthesis. This is in bacteria, by the way. And right there we have the reactions. So the elongation is going to begin with the formation of our acetyl ACP and the malonyl ACP. It's going to be catalyzed by the acetyl transacylase and the malonyl transacylase. The intermediates are going to be attached to the ACP and these reactions. So here we have the acetyl-CoA plus the ACP. We're going to swap the acetyl onto the macro-CoA, or the ACP, right? And then we're going to take malonyl-CoA, we're going to swap it onto the ACP, and those were equilibriums. The synthesis of the odd number fatty acids is actually going to begin with the propionyl ACP. This is going to be formed from the propionyl-CoA by the acetyl transacylase. And again, if you want to make an odd one, you have to start off odd, right? Because you're going to be basically adding like the beta oxidations in reverse. Okay, so 2, 2, 2, 2. Okay. The acetyl ACP and the malonyl ACP are going to be condensed to form the acetoacetyl ACP, catalyzed via the beta ketoacyl synthase. This is your condensing enzyme. This is the one that does that condensation we started with in the previous pathway where we compared the two. The acetyl ACP plus malonyl ACP gives you the acetoacetyl acetyl ACP plus a freed ACP plus CO2. Again, in many ways, you can actually just go, I don't care, CoA ACP, or just go macro CoA versus CoA, right? Though in these cases, you do use ACP. Okay? But it serves essentially the exact same purpose. It's just we're doing this in the cytosol. So we need to use the macro CoA, the ACP. The acetoacetyl ACP is going to be reduced to the D3-hydroxybutyryl ACP. This can be catalyzed by the beta-ketoacetyl reductase. NADPH is actually the reducing agent. This is one of those places where this really diverges a bit because instead of using NAD and FAD, we use NADPs, both places. So the D3-hydroxybutyryl ACP, this is going to be dehydrated, excuse me, form the crotonil ACP, catalyzed by the 3-hydroxyacyl dehydratase. Crotonil ACP is going to be reduced to the butyryl ACP, catalyzed by the enol reductase, and NADPH is going to be your reducing agent in that reduction. Notice again, this is the D3-hydroxybutyryl. So in the second round of your fatty acid synthesis, your butyryl ACP is going to condense with malonyl ACP. This will form your C6 beta-ketoacetyl ACP. Reduction, dehydration, and a second reduction will convert the C6 beta-ketoacetyl ACP into the C6 acyl ACP. The elongation will cycle, continues until we get to a C16 acyl ACP, and that's going to be formed. And notice that's sort of the limit of large chain, and so we get that large chain, right? This is going to be your saturated acid, fatty acid synthesis, and this just sums up that same reaction we just looked at. That's all it is, okay? Into a single pathway instead of text. So the fatty acid synthesis reactions are going to be similar in E. coli and eukaryotes. In animals, the enzymes required for the fatty acid synthesis are actually linked in a single polypeptide chain. The mammalian fatty acid synthesis is going to be a homodimer with two distinct compartments. Each subunit possesses all the necessary enzymes. Each non-catalytic Poseidon domain, are going to be maintaining enzyme structure. Are you all of you in frats and sororities? Or is anybody not in a frat and sorority? Okay, so half-half. Okay. Do you still have to memorize the Greek lettering? Oh. Oh, okay. Okay. Oh, wow. Okay. The ladies are just kicking the shit out of you guys for their requirement levels, aren't they? No, maybe you guys have some other requirement that's rough.
Yeah, we make up for it because there's a whole bunch of other memorization
Fair enough.
stuff, so it makes us.
Fair enough. Okay, so this is again that mammalian fatty acid synthase right here. Again, large dimeric multi-domain protein complex. Notice again, it's basically mirror images because it's a homodimer. Everybody said okay? And that does that full synthesis process in mammals. We have the whole thing on one. So one catalytic cycle for mammalian fatty acid synthesis is going to look something like this. So here we have the ACP, and right here we have our acyl group right there. And so the first thing that's going to happen is we'll have the KS domain, this is going to have this sulfide link onto the new acetyl group right here. Notice that this one's going to have that carbonate that we talked about for the beginning of that reaction, right here. Okay, and then this is then going to link those two. That's going to be that joining, that condensation. And then we'll have that chain right there. Then we'll go on to the next step. And this is going to have, notice we had the carbonyl in this step. That's going to go to the alcohol. Again, that's going to be the D form. Then what's going to happen, that's the Kr down here, by the way. The next step is going to go to this DH domain. That one's going to be where we get the carbonyl right here, going to the double bond right there. Okay. The next step, bless you, is going to go over to this ER. Notice that we're actually going what direction? Clockwise. Yeah. See that? You just clockwise through this. Okay. Okay, and so we have this right here where we have the carbonyl is going to be reestablished right there. And that's going to go on in the ER portion, the domain. That's going to have your linkage to your next group right here, which is going to have the KS. Nope, that is this right here. Very, very similar, right? Everybody see the similarity? Okay. Again, that thiol is going to connect to an acyl group. And that's where we get those linkage with our new carboxylate right there. Okay. And then we're going to go back onto the KS because we already had linkage. So, round and around and around, and we keep linking more and more carbons on there, right? Until what? We get to C16. And then we stop, release that. Congratulations, you made your fatty acid. Okay? You know what? I actually don't remember. I believe it does, if I remember correctly, but I'd actually want to look that up and reconfirm. Because I don't remember if it absolutely has to stop at C16. I may have put something in here, but if I did, I don't remember off the top of my head. Yeah, hopefully I did. If not, I believe it does, but remind me and I'll look it up, okay? Your cycle is going to begin after your malonyl acyl transacylase, your MAT. This moves an acyl unit from your coenzyme A to the acyl carrier protein. That's that first equilibrium that we talked about, where we transfer onto the macro-CoA, the ACP. The beta keto synthase, this is your KS, by the way. So that's right there, KS. This is going to be the unit going to accept the acetyl unit. The MAT is going to be what attaches a malonyl unit to the ACP. Okay? And so that's this right here, the KS that we just referred to. So your malonyl ACP is going to deliver a malonyl unit to the active site of your KS. This is going to form the beta ketoacyl product. This is going to be attached to your ACP. Your ACP is going to deliver the beta ketoacyl product to the keto reductase. That's the KR. That was the next one right there, the yellow one. Over here. Okay. And then, again, keto reductase. This is then going to reduce your beta keto form group to the OH, and that's where we had that chirality form and we have the D isomer, okay? Stereochemistry. All right, so the step 4 is going to be the beta hydroxy product. This is going to be delivered to the dehydratase, DH. This is going to introduce a double bond with the loss of water. Again, the dehydration. And again, that's this DH down here, which is your green unit right there. Okay. All right, and then the step 6 is going to be your ACP delivers the reduced product to the KS. This will then be recharged with a malenyl-CoA, and this will be done by that mat, and that's over here. All right, and KS is then going to condense your 2 molecules on your ACP. This is the stoichiometry for the synthesis of palmitate. Right there. And again, note we have the acetyl-CoA plus 7 of these, meaning a total of 8 times 2, which is your 16. Okay. The synthesis of the required malenyl-CoA is this right here. Your overall stoichiometry for the synthesis of palmitate is that. Note again, we use NADPHs alone, and we use 7 ATPs to power this process forward. So your acetyl-CoA must be transferred from mitochondria to the cytoplasm for fatty acid synthesis. The mitochondria are not readily permeable to the acetyl-CoA, and so you wouldn't have free flow of the reactants that you're going to be using a bunch of, right? And so it won't make any sense to do it in there. The citrate is formed from the acetyl-CoA and oxaloacetate in the mitochondrial matrix and can be transported to the cytoplasm. The ATP citrate lyase cleaves citrate into acetyl-CoA and oxaloacetate. And this is this reaction, citrate plus the ATP plus the CoA plus water, acetyl-CoA, ADP, orthophosphate, oxaloacetate. This right here is going to be that transportation. And again, the acetyl-CoA, this can be transferred mitochondrial matrix indirectly, but it's going to take that citrate and pyruvate exchange. Okay? And so again, mitochondrion right here, acetyl-CoA goes to the citrate. Citrate is then exchanged for pyruvate into the cytoplasm. Cytoplasm is citrate is going to go to acetyl-CoA and oxaloacetate. NADH needs to be used to give us malate. This is going to give us NADPH and pyruvate. Pyruvate is the one that was exchanged for the citrate, coming back into the mitochondrion. And this is going to give us oxaloacetate, normal pathway. I know. Weird, right? Yeah. Remember that a lot of this stuff has to do with the fact that even though it may seem like it's inefficient, when you're talking about these 3 formed carbon groups being exchanged around, we're not losing that much energy because we're not losing that much carbons, that many atoms. On the other hand, if we were to have to duplicate these pathways to exchange stuff over and over and over, how much carbons and atoms would we lose and how much energy would that take? Okay? That's one of the sort of things you have to kind of keep in mind. Okay? Is the machinery has a cost. Right? The inner mitochondrial membrane is impermeable to oxaloacetate. A series of bypass reactions are going to generate most of the NADPH needed for fatty acid synthesis. The malate dehydrogenase is going to catalyze the reduction of the oxaloacetate to malate by NADH. The oxaloacetate, NADH plus H plus, gives us malate, NAD plus. Malic enzyme is going to catalyze the oxidative decarboxylation of your malate to your pyruvate. This is going to yield your NADPH, and I'm just talking about the pathway I showed you before. The pyruvate is going to enter the mitochondrion. Pyruvate carboxylase catalyzes the carboxylate of the pyruvate to oxaloacetate. This will give you this overall reaction. Your sum of your 3 reactions is this right here. And that's the overall reaction. Note, by the way, because you get exchange back and forth, and it ends up creating a cycle, your overall reaction, the sum of those reactions, is just energy changes. Okay? And so you just have your NADPs, NADHs, ATPs, energy changes. Okay? your transporters. One molecule of NADPH is generated for each molecule of acetyl-CoA that's transferred from the mitochondria to the cytoplasm. The additional NADPH is synthesized by the pentose phosphate pathway. My God, everything links. Yeah, you'll spend the next 20 years making all these links in your brain. You'll be like 50 years old and you'll be going, damn it, Dr. Clausen, I get it now. Okay. The glucose, again, pentose phosphate pathway, ribulose 5-phosphate. This gives us the NADPH. Right here, we actually see the NADPH coming off of this citrate, going to oxaloacetate, going to melodate, going to pyruvate. Remember, glucose through glycolysis can actually feed into this right here, which is the pyruvate. The acetyl-CoA right here from the citrate can actually go with NADPH and form fatty acids. That's what we just added to this. This right here is that mitochondria, which you may recognize from your citric acid cycle. Big hint being citrate right here. This is the same thing that can then exchange outwards. And this is that exchange pathway going to pyruvate. The cancer cells are going to increase fatty acid synthesis for use as signal molecules and for incorporation of new cell membranes. Many fatty acid synthesis enzymes are going to be overexpressed in most human cancers. The expression is correlated with tumor malignancy because it's spreading then. The inhibition of beta-ketoacyl ACP synthase is going to inhibit phospholipid synthesis and subsequent cell growth in some cancers by inducing apoptosis. Mice treated with inhibitors of beta-ketoacyl ACP synthase ate less and showed remarkable weight loss. Inhibition of acetyl-CoA carboxylase, ACC, in prostate and breast cancer cell lines actually induced apoptosis in the cancer cells, which is quite impressive, actually. Really cool. Triacylglycerols are going to be promising materials for renewable biodiesel fuels. Syngas, this is a collection of CO2, CO, and H2. I don't know if biodiesel fuels is actually going to be a thing that happens in the future. It's probably going to end up being feedstocks, actually. But they're still really cool and interesting stuff. The acetogenic bacteria, this is going to be bacteria that contain the wood lungdel. I totally mispronounced that, but you can fix that sometime by looking his name up. Pathway for synthesizing acetate from syngas. Syngas captured from municipal waste or other waste sources. These are fed to anaerobic acetogenic bacteria. The acetate can be harvested and used as carbon source for oleaginous yeast. These can synthesize and accumulate triacylglycerols for things like biofuels and other renewable products. I guess I'm actually out of time, so we'll stop here. And I will see you all Friday.